So now we're going to start about telling you all about our latest research. Some of it's ongoing and some of it is starting up, and there's a few I think there towards the end as well. So we'll first be hearing from Dr. Rabinovici. So Dr. Rabinovici received his BS degree from Stanford University and his MD from Northwestern University Medical School. He completed a neurology residency at UCSF and a behavioral neurology fellowship at the Memory and Aging Center here. Dr. Rabinovici is now an assistant professor at UCSF. His research focuses on how structural, functional, and molecular brain imaging techniques can be used to improve diagnostic accuracy in dementia and to study the biology of neurodegenerative disease.
Thanks, Christine. Thanks, everyone, for coming on this rainy day. Blessedly rainy, I'd say, for the Bay Area. As we heard from both Robin and Bruce, while neurologists can suspect the diagnosis of PSP or CBD, the definitive diagnosis isn't really made until the time of autopsy. And what the pathologist is looking for, and Bruce showed some images of this first, is these accumulations, these black things that you see here of the tau protein. Now, the tau protein is normally present in our brain cells, and as a single unit, it stabilizes the skeleton of neurons, of brain cells. But what happens in both PSP and CBD, as well as other diseases, is that tau becomes a sticky protein. One unit sticks to another, and eventually you see these we think toxic accumulations occurring in neurons or in astrocytes, which are support cells in the brain. And you can see what they look like. Some of these inclusions are typical of PSP and CBD. Others are more typical of PSP. But this is what the pathologist is looking for when they look under the microscope. Now, why can we as neurologists infer that tau protein is present in the brain? Well, in particular diseases, the tau protein tends to affect particular brain areas that have specific functions. And so zooming out here, you see all this brown stuff is tau protein and CBD accumulating in the frontal lobe of the brain, regions of the brain that are incredibly important for cognitive control, for behavior, for planning movement. And when tau accumulates in this area, you get dysfunction of the brain cells, you get the symptoms of CBD. And conversely here, you see brown things. This is tau protein accumulating in the midbrain in areas that are incredibly important for eye movement control, leading to the symptoms of PSP. And so what I want to talk to you about today is a very exciting new advance in brain imaging that allows us to detect these tau inclusions and quantify them not at the time of death, but actually during life using a brain scan. And so how does this work? Well, I'll talk to you today about two small molecules, and for those of you who had organic chemistry, here are the structures. One is called T807, the other is called PBB3. But the key thing is these are small molecules that we can actually inject IV. They get into the brain. They stick to tau protein. Oops. Managed to lose the signal here. Oh. And they have a radioisotope labeling them. So carbon-11 or fluorine-18, this is an isotope that emits a radioactive signal. And what you see here fluorescing are tau proteins labeled with one of these molecules, PBB3, and this sends out a radioactive signal that we can detect with a PET scanner. And then the PET scanner can actually figure out where the radioactivity is coming from, and you can reconstruct that into a three-dimensional image. And what you see here in red and yellow is areas in the brain. This is a PET picture of the brain now, where radioactivity is coming because tau protein is present in the brain. And I think this technology really has the potential to revolutionize not only our ability to make an early and accurate diagnosis of PSP and CBD, as Bruce was referring to, but actually to develop treatments targeting the tau protein. And I think we'll hear more about this from Adam. So here's some work that we've done in collaboration with investigators at the University of Pittsburgh. And the first thing we wanted to know is, do these small molecules, in this case T807, can they bind to tau protein in Alzheimer's disease? So Alzheimer's disease also involves the tau protein. Tau protein is a supervillain in a lot of neurodegenerative diseases. And here you see a tau inclusion in Alzheimer's disease. It looks a little bit different than what we see in PSP and CBD, and in fact it accumulates in different brain areas, initially in areas that affect memory, thus the memory loss in Alzheimer's. What you see in this picture is this small molecule, T807, binding to tau protein in Alzheimer's. In red, you see an antibody binding to the same tau proteins. Now, antibodies are great at binding to specific proteins, so you might ask, well, why don't we just use antibodies to image these proteins in the brain? It turns out that antibodies are too big to get into the brain. The brain has a defense system called the blood-brain barrier that prevents large molecules from getting into the brain. But what you see in purple here is that the small molecule that does get into the brain shows the same thing that the antibody does. Here in purple is the convergence. We are able to bind tau protein in Alzheimer's disease. And here are some pictures of brains. This is a healthy volunteer. This is a patient with very mild Alzheimer's. You see some green, some early tau deposition in the temporal lobe. Here's a patient with mild Alzheimer's showing more yellow and red now. And here's someone with severe dementia due to Alzheimer's disease. You see a lot of accumulation of tau protein. So next we wanted to ask, well, these small molecules bind in Alzheimer's disease. Do they also bind to tau in other diseases? And the answer is yes. And so here is more work. This is, again, work that was done with brains donated by our patients. So this is another illustration of what kind of advances we can make with brain donations. Here you see this small molecule again, T807. All these little blue dots are T807 binding to tau inclusions in Pick's disease. This is one of the diseases that Bruce mentioned as a tau disorder. This disease causes frontotemporal dementia usually. Here you see a low magnification and high magnification. And here's again the antibody stain showing that the small molecule is recognizing the same sort of tau deposits that you can see with an antibody. And here's PSP. This is a tau inclusion called a tufted astrocyte. And you can see at low and high magnification that this molecule is avidly binding to the same things that we can see with the antibodies. So this looks like a very promising way for imaging tau in PSP. Here's another small molecule that we're looking at called PBB3. And again, in green this time, you can see PBB3. All these fluorescent areas are PBB3 binding to tau protein here in CBD and here in PSP. Again, binding to the same areas that we see in red with the antibodies. And here's the nice picture that I showed you earlier. This is courtesy of our collaborator, Makoto Higuchi in Japan, who developed this particular imaging molecule, PBB3. So how does this look as a brain image? Well, we received this slide courtesy of our collaborator, Brad Dickerson at Harvard. This is the brain scan of the first patient in the United States with PSP to have a tau brain scan. And what you see here are areas where the tau, where the image showed high signal compared to background. And these are very interesting areas. You see high signal in the midbrain, again, an area that's very important for eye movement control, for motor control of the body and extremities. You see signal in the thalamus, which is a critical relay station in the brain. And you see signal in the dentate nucleus of the cerebellum. This is an area that's very important for balance and coordination. And the key thing here is the signal that we see on the brain scan corresponds very nicely with the areas that we know develop tau deposition in PSP. So this is a very promising first scan. Our collaborators in Japan have studied 9 patients with PSP and 3 with CBD. And here you can see the summary of their results. This is in green the areas that they saw signal in PSP. So again, the midbrain, this area called the basal ganglia, which is very important for motor control and can lead to some of the Parkinsonian symptoms that we see in PSP. And in contrast, in CBD, they saw signal in the frontal lobe, again, an area important for motor planning, cognitive control. So again, the signal that was seen on the brain scans corresponds very nicely with where we expect the tau pathology to be in the brain. So these are now not postmortem samples. This is imaging tau in the living brain. So how can this help us develop drugs? Well, the first thing is that tau PET scans can help us identify those patients that have tau in their brain at an early stage, those patients that are most likely to benefit from a drug that can stop the spread of tau through the brain. Now, this is less of an issue with PSP. If the diagnosis of PSP is made during life by an expert, about 90% of the time you will find the tau protein in the brain. But in corticobasal syndrome or corticobasal degeneration, the neurologists are less accurate. About 60% of the time, the tau protein will be found in the brain, and for this reason, actually, many companies that were thinking of doing clinical trials against the tau protein didn't want to include CBD patients in those trials because they felt that the clinical diagnosis wasn't reliable enough. Well, if we have a scan that can detect and confirm the tau protein, those patients should now be able to enter the trials. You can see if your drug's a winner very early. You can see if it's lowering the levels of tau in the brain by scanning patients before and after treatment. You can see if the drug is slowing the spread of tau through the brain. So you get a good early sense if there's a biological effect of any drug that you're testing. And the ultimate vision is that these scans will allow very early detection and intervention. And this may sound like science fiction now, but this is exactly the direction the Alzheimer's disease field is moving, using a different kind of PET scan, an amyloid PET scan, that detects one of the proteins that accumulates early in Alzheimer's disease. And so we're excited to announce that we are going to offer these PET scans to our participants beginning this spring. We're going to do this in collaboration with Lawrence Berkeley National Laboratory in Berkeley. So we will ask people to go to Berkeley for these scans. And initially, we'll probably ask people to have 2 PET scans, one to look for the tau protein and another to look for the amyloid protein. That's a protein associated with Alzheimer's, but also with aging. And it's really going to be important for us to get both scans to get a full understanding of what's going on in the brain at the molecular level. We'll ask people to come back once or twice more to see how the accumulation of tau is changing over time. And as we'll hear from Adam, we're very excited to offer these kinds of scans as part of the clinical trials that we'll be starting in order to see if the drugs are having an effect not only on symptoms, but on the amount of tau that we can measure on a brain scan. And so I think this is a very exciting time because this real major advance in our diagnostic and imaging tools goes hand in hand, I think, with developing and testing new drugs. So I'll end there. I'd like to thank Bruce Miller and Adam Boxer, of course, here at UCSF, our collaborator Bill Jagist at Berkeley, our collaborators at Pittsburgh as well, and the initial funding for some of these pilot studies will be supplied by the Tau Consortium. So thanks very much.